Welcome to CD Bioparticles channel. In this video, we'll present you magnetic particles for cell-free DNA extraction. Cell-free DNA, cfDNA, are nucleic acid fragments that circulate in plasma, serum, and other bodily fluids outside of cells, first discovered in 1948 by Mandel and Metes. Cell-free DNA is present in healthy people in varying, usually small, amounts. Conditions which increase inflammation such as systemic lupus erythematosus, arthritis, and hepatitis are associated with increased higher total levels of cfDNA compared to healthy controls. In 1977, Leon et al. Discovered that cancer patients had higher levels of cfDNA in serum, with significantly higher levels in patients with metastatic disease as compared to non-metastatic disease. In the past several years, there has been increasing enthusiasm for the use of cfDNA as a biomarker for cancer, with uses from diagnosis to prognosis to monitoring tumor evolution and response to therapy. The ability to detect and quantify tumor mutations from a blood sample, so-called liquid biopsy, holds great promise to revolutionize cancer management. Two possibilities exist for the main origins of cfDNA: cellular breakdown mechanisms, such as apoptosis and necrosis, and active DNA release mechanisms, such as exosomes, vertisomes, and argonaute. Tumor-associated DNA is primarily derived from apoptosis of primary, metastatic, and circulating tumor cells, CTCs, as well as active release from proliferative tumor cells, which is known as circulating tumor DNA, ctDNA. In general, ctDNA only accounts for a small fraction of the total cfDNA, and there is no way to isolate ctDNA, especially from other cfDNA. Only the emergence of tumor-related molecular alterations in cfDNA indicates the presence of ctDNA. cfDNA testing, also known as liquid biopsy, is a powerful diagnostic tool that analyzes fragments of DNA found in a person's blood. It has various applications in the field of medicine. Here are some important applications of cfDNA testing. Cancer detection and monitoring. Cancer patients usually have a high level of cfDNA in their serum or plasma as a result of cellular necrosis or apoptosis, because tumor cells divide faster than normal cells and cfDNAs are released in a high proportion. The fraction of cfDNA that derived from tumor cells is named circulating tumor DNA. In recent years, both cfDNA and ctDNA have gotten huge attention as novel blood biomarkers, as quantification and kinetic analysis of cfDNA and molecular profiling of ctDNA have suggested their predictive and prognostic values. Several liquid biopsy tests, designed for the identification of cancer-specific mutations, have been recommended as companion diagnostic tests by the European Medicines Agency and Food and Drug Administration of USA to guide therapeutic decision-making. Such tests include the COBAS EGFR mutation test for non-small lung cancer or BRCA-analysis CDX for breast and ovarian cancer. The applications of cfDNA in cancer are shown in figure. Prenatal testing. cfDNA testing is commonly used in prenatal care to screen for certain genetic conditions, such as Down syndrome, Edwards syndrome, and Patau syndrome. By analyzing fetal DNA in the maternal blood, cfDNA testing can provide accurate and early detection of these chromosomal abnormalities without the need of invasive procedures like amniocentesis or chorionic villus sampling. Diagnosis of infectious diseases. cfDNA testing can identify a wide range of pathogens, including bacteria, viruses, fungi, and parasites. This comprehensive identification can aid in determining the appropriate antimicrobial treatment and help prevent the unnecessary use of broad-spectrum antibiotics. Last but not least application: transplant rejection monitoring. Following organ transplantation, cfDNA testing can be employed to monitor graft rejection. By analyzing the cfDNA in the recipient's blood, it is possible to detect the presence of donor DNA, which indicates potential graft rejection. This non-invasive method allows early intervention and adjustment of immunosuppressive therapy, improving transplant outcomes. We extract cfDNA because it offers a non-invasive way to obtain genetic information. By analyzing cfDNA, we can detect diseases like cancer, monitor treatment response, and screen for genetic disorders like Down syndrome. However, extracting cfDNA presents several challenges. The primary challenge is the low concentration of cfDNA in bodily fluids. This requires sensitive extraction methods to isolate sufficient amounts for analysis. Another challenge is the short fragment length of cfDNA, which can make it difficult to amplify and sequence. Additionally, the presence of other cellular components and contaminants in the sample can interfere with the extraction process. To overcome these challenges, researchers have developed various techniques, including magnetic bead-based and column-based methods, to efficiently extract high-quality cfDNA. Here is a comparison of cfDNA extraction methods. Magnetic particle-based extraction stands out due to its short operation time, no need for multiple centrifugation steps, automation compatibility, ability to capture small fragments, and fragment screening capability. Silica gel column-based extraction is also relatively quick and can capture small fragments, but it requires multiple centrifugation steps and is not as easily automated as magnetic particle-based methods. Chlorophenol, high-intensity salt precipitation, and ion exchange methods are generally slower, require multiple centrifugation steps, and are not easily automated. They also cannot capture small fragments or screen for specific fragments. Magnetic particle-based cfDNA extraction is a powerful technique that leverages the properties of magnetic beads to isolate cfDNA from biological samples. Here's a breakdown of the process. 1. Magnetic bead preparation. Magnetic beads are synthesized with a core of magnetic material, often iron oxide, encapsulated in a silica matrix. The surface of these beads is functionalized with specific groups, such as silanol or amine groups, which have a high affinity for nucleic acids. 2. Sample preparation. Biological samples, such as blood plasma or serum, are typically treated with lysis buffers to release cfDNA from cells. 3. Binding of cfDNA to magnetic beads. The lyse sample is mixed with the magnetic beads. The functionalized surface of the beads binds to the negatively charged phosphate backbone of cfDNA through electrostatic interactions. 4. Magnetic separation. A magnetic field is applied to the sample, causing the magnetic beads bound to cfDNA to aggregate and form a pellet. The supernatant, containing unwanted cellular debris and other contaminants, is removed. 5. Washing. The magnetic bead cfDNA complex is washed with buffers to remove any unbound contaminants. 6. Solution. A low-salt buffer is added to disrupt the binding between the cfDNA and the magnetic beads. The purified cfDNA is released into the solution. 7. Recovery of cfDNA. The magnetic field is removed, and the purified cfDNA can be collected for downstream analysis, such as PCR or sequencing. So, to sum it up, magnetic particle-based cfDNA extraction really shines compared to traditional methods. First, it's fast and efficient. The magnetic beads quickly grab onto the cfDNA and release it when needed, saving time. Second, it's super clean. These methods effectively remove impurities, ensuring we get high-quality cfDNA for further analysis. Third, they're highly adaptable. You can automate the process, making it perfect for large-scale studies. Plus, they work with a variety of sample types, giving us flexibility in our research. Fourth, they're sensitive. Even tiny amounts of cfDNA can be captured, which is crucial for early disease detection. Lastly, they're reliable. You get consistent results, no matter how many samples you're processing. Overall, magnetic particle-based cfDNA extraction is a game-changer, making it easier and more accurate to study cfDNA and its potential applications in medicine. Magnetic particles have revolutionized the way we isolate and analyze cfDNA. But how can we make them even better? One area of intense research is the development of novel materials. By exploring materials like cobalt ferrite and manganese ferrite, we can create particles with improved magnetic properties and biocompatibility. Another exciting development is the integration of magnetic particles with microfluidic devices. These tiny, fluid-filled channels allow us to perform complex biological assays on a very small scale. By combining the power of magnetic particles with the precision of microfluidics, we can develop rapid, sensitive, and automated cfDNA analysis platforms. Imagine being able to diagnose a disease with a simple blood test, right at your doctor's office. This is the goal of point-of-care testing. Magnetic particles are playing a crucial role in making this a reality. By capturing cfDNA from a small blood sample, we can quickly and accurately detect a wide range of diseases. The future of cfDNA analysis is bright. As we continue to advance magnetic particle technology and integrate it with emerging technologies, CD Bioparticles provides cfDNA magnetic silica particles and extraction and purification kits for isolation of cfDNA from whole blood, serum, plasma, urine, and other body fluids. Our products are used for rapid extraction of cfDNA from 0.5 to 10 milliliters serum and plasma samples and can meet the requirements of automated and large sample volume operation in NGS, PCR, and other molecular biology experiments. Thanks for watching. For more information, please visit www.cd-bioparticles.com.